So just to give a quick reminder about DNA replication, so if you look at this portion of the DNA molecule here, we've got our double-stranded DNA. This is just a picture illustrating from your book the process of DNA replication. So we have our helicase, DNA B, that's the one that's unwinding the DNA, and you can see that it's going in this direction. This is our topoisomerase here, or DNA gyrase, that is helping relieve the supercoils, our single-stranded binding proteins, our DNA polymerase 3, that's the major DNA polymerase. And as if you look down here on the bottom, it gives you that full structure of the DNA polymerase where it has the beta clamps that are clamping it to the DNA, the polymerase portion, which are these little guys that kind of look like hats or Santa hats or something like that. That is the actual polymerase part. So on this picture in the front here, you can see the little green molecules are the primases, and that was added by, or the primers that were added by primase. And now once the primer is on there, the DNA polymerase can come in and add the DNA. So we've got on this side our leading strand, and that leading strand can continue on as DNA helicase opens up the DNA. This side of the polymerase can keep going. But as we see on the lagging strand, it's having to do this in parts. And again, that's because the direction of the DNA polymerase can only replicate it in that 5' to 3' direction, which is works perfectly fine going this way on the leading strand, but on the lagging strand has to go in the reverse direction. And so as primase comes in and adds a primer, the DNA polymerase comes back in and can continue adding strands of DNA. So we get these short fragments of DNA we call the Okazaki fragments. Now in reality, as I mentioned last time, when we're looking at the holoenzyme and the enzyme together, we can see that it's for the lagging strand to be correctly replicated, it's actually folding up into this configuration so that the enzyme can work on both the leading and lagging strand at the same time. So here we show a little bit more detail on the DNA polymerase in that conformation. So we have the core polymerase here. We've got our beta clamps here. Our leading strand is going this direction, and our lagging strand is going this direction. And you can see that in that loop that we have both the leading strand and the lagging strand going in the same direction. So by looping the DNA around, we're actually able to have both sides go through the DNA polymerase in the correct orientation so that the polymerase is adding DNA in a 5' to 3' mechanism. And this just shows you the primase adding the primer there, so the primase comes in and adds the primer, so then the DNA polymerase is able to make DNA off that primer again. Once the DNA polymerase runs into the next Okazaki fragment here, so you can see that it made DNA all the way through here, runs into the next Okazaki fragment, has to release the DNA and reload back onto the DNA so that the clamp loader comes and loads it onto the next piece of DNA so that the lagging strand can come and continue making DNA. And once that moves into position, the beta clamp is left behind, the DNA polymerase can come in and continue making DNA. So once this process goes on over and over again, we have both the leading and the lagging strand. We're able to come back in there, remove the primers from the lagging strand, which is not shown on any of these slides. The primers are removed, the DNA ligase comes in and is able to ligate the fragments together, and we have two complete pieces of DNA. Okay, with that, we're going to move on to talking about DNA repair. So let's first talk about DNA mutations. So some chemical reactions and some physical processes in the cell will damage genomic DNA. There are thousands of lesions per day, but most of those mutations are repaired thanks to the DNA repair system. So even though there is lesions in the DNA, they only turn in mutation 1 in 1,000 times thanks to this DNA repair. In the human genome, there is about a little over 130 genes for repair proteins. So this is a Accumulations of mutations in those eukaryotic cells correlate strongly with cancer, showing that some substances that cause DNA mutations, which are called carcinogens, mutinize the DNA, leading to cancer. The majority of the mutations that occur that are repaired in the cell is corrected by using the undamaged strand as a template. If some bases change and they escape the repair process, that incorrect base could serve for template for replication from then on. So the majority of the mutations that happen, as long as we've got the undamaged strand available, we correct that mutation very easily. If somehow that base change escapes repair, then that could be a mutation that's permanent and goes on to the daughter cells during replication. And that will be a daughter cell that carries the change sequence in the full strands. So let's talk about the types of damage that can be done to DNA. So mismatch repair, that can be a repair where A's no longer bind with T's or G's no longer bind with C's, that there is a mismatch in there somehow. And that's from incorporation of the wrong nucleotide. There could be abnormal bases that arise from spontaneous deamination, alkylation, or exposure to free radicals, so actually damaging the base of the DNA. The pyrimidines can form dimers, and this happens when they're exposed to UV light. Or there could be lesions in the backbone, so actual tears in the backbone of the DNA, and this is caused by exposure to ionization radiation or free. So when the DNA repair systems do repair of mismatches, how does the repair enzyme know which strand is the correct strand? So we have a mismatch. Obviously, one of the nucleotides is incorrect. How does the repair system know which one is correct? Well, in E. coli, the parent strand is methylated. So if we're talking about in the process of DNA replication, the strand that's used as the template is methylated in order to mark it as that parent strand. So an enzyme called DAM methylase inserts a methyl group at adenine, so at the A's, in the sequence of the DNA. So during replication, the newly synthesized strand remains unmethylated for a short period of time after synthesis. So if there's any sort of mismatch, we have our parent strand and our newly synthesized strand, and you can see we have a little mismatch here. The parent strand will have these methyl groups on it. I apologize for my writing on the computer. And so that the repair enzymes will know that that is the parent strand. So this one has the correct base, and this strand right here is the one that has the incorrect base and is causing the mismatch. So any of the replication errors must reside in the unmethylated strand, like I just said. I'm sorry for that. Maybe I can delete that. Hold on. There you go. The methyl-directed mismatch repair system will then cleave the unmethylated strand, so the one that has the mismatch, and initiate the repair process. So if we look at that in pictorial representation, so here we have our original strand here, our parent strand, both the top and the bottom are methylated. You can see the methyl groups right there. And during replication, that parent strand remains methylated. So we've got the methyl group, and that our new strand here, the pink one, is unmethylated. So at the point of replication, we can see that it's hemly methylated, and saying that the old strand, the parent strand, is methylated where the new one is not. After a while, DAM methylates will come in and continue to methylate the new strand. But at any point during this hemimethylated state that there is a mismatch, the mismatch repair system will know which one is the parent strand. So let's talk specifically about mismatch repair. So in mismatch repair, we have two enzymes called MutAL and MutS, and they're responsible for recognizing the methylated DNA. So here we have our mismatch. Okay, this is our new strand. This is our old strand. We can see that our old strand is methylated. Mute S and mute L are able to bind to the DNA and then look for this methylated strand. Okay, so we have the complex of MUT-L and MUT-S bound to the DNA here, and the next protein, MUT-H, comes in and binds to the complex. That complex is able to recognize the methylated DNA and create this loop structure where the mismatch is located. The MUT-H then cleaves the non-methylated DNA strand, so the one that's incorrect, on the 5' side of the guanine. So, like I mentioned, there is GATC location of those methyl groups at that guanine, so that's probably like right there. Of the unmethylated one, it becomes cleaved, so right there. The DNA unwinds and is degraded in a 3' to 5' method. Helicase 2 single-stranded binding proteins and an exonuclease work together to degrade the non-methylated DNA. So we can see the DNA is degraded here. Which exonuclease that's used, so there's different exonucleases. We can see there's 1, 5X, RecJ, depends on the location of the cleavage site relative to the mismatch. The removal of the sequence, so we've had the sequence removed here by the exonuclease, is then replaced by DNA polymerase 3, and then DNA ligase will ligate it back together, so it's a perfectly whole molecule at the end of the mismatch repair. So that was mismatch repair. Let's talk about base excision repair. So this is removal of a base. So removal of the base happens from DNA glycosylases. And glycosylases are able to recognize specific lesions and cleave that N-glycosyl bond between the sugar and the base of the nucleotide. So if we're like, oops, erase that, try that again. So if we're looking at the sugar, and here's your phosphate group of your nucleotide, and then here would be your base. This is cleavage of this bond here, so between your sugar and your base. Okay. What does that create? It creates sites or nucleotides that don't have the base, so antipyrimic or antipyridinic sites, or AP sites for short. For example, there is a uracil glycosylase that removes uracil from DNA. So if you remember back when we first talked about DNA last time, we know that uracil is not in DNA. DNA has A's, G's, T's, and C's, and that U is only replaces the T in RNA. So if we see uracil in DNA, that means it shouldn't be there. And that happens because cytosine spontaneously deaminates to create uracil. And uracil glycosylase can come in there and remove the base. And this happens 100 times faster in single-stranded DNA than it does in double-stranded DNA. As we know, the double-stranded DNA is more stable. So depending on what glycosylase is out there, it'll make these minus base sites, these antipyrimic or pyrimidinic sites, by hydrolyzing that bond between the sugar and the base. So repair of these antipyrimidic or pyrimic sites in bacteria, the entire nucleotide is ultimately removed, just not the base. So even in fact, some of that region around the AP site is also removed. So these AP nucleases cut the DNA backbone around that site with the base removed and removes the DNA. DNA polymerase 1 comes back there and synthesizes new DNA. DNA ligase comes in and seals the next. So if we are looking at a uracil occurring in DNA and it's making a mismatch here, DNA glycosylase and specific uracil glycosylase will remove that uracil. The AP endonuclease will remove the nucleotides not only right where the mismatch was and the wrong base, but a section around that. DNA polymerase 1 will come back in there and replace the DNA. DNA ligase will seal up the nick, and we'll have fully a new strand with the correct base in it. We just talked about base excision repair. Now let's talk about nucleotide excision repair. So there can be large distortions or large damages to the DNA that needs to be repaired, and that's repaired by the mechanism of nucleotide excision repair. These lesions include things called pyrimidine dimers that I talked about before. That's pyrimidines that will form dimers together, and that's caused by UV light, and other large damages caused by either UV light or cigarette smoke. So these are huge distortions in the DNA that are caused by some carcinogen. So the pathway involves removal of the DNA segment by nucleases. So if we're looking at a DNA lesion here, and this is, I think, supposed to represent a pyrimidine dimer, and we're looking at both E. coli and human nucleotide excision repair. So the exonucleases come in there and remove a section of the DNA, including that damage. So we have this huge piece. That also occurs in humans. They are called ABC nucleases in E. coli, and they contain UVRA, B, and C. They hydrolyze the 5th bond on a 3' side of the lesion and the 8th bond at the 5' side of the lesion. So there's a 13 nucleotide segment, or a 13er, that is removed from the DNA, a little bit larger in humans. DNA polymerase will come in there and fill in the gap. DNA ligase ligates it together. Similarly, in humans, one of the DNA polymerases comes in and adds new DNA, and the DNA ligates them back together. And again, the nucleotide excision repair is for these large DNA lesions like pyrimidine dimers. Just giving you the words to what I talked about in humans. So again, different polymerase comes in and ligates them. So that is just a brief explanation of DNA repair and the different DNA repair mechanisms. Now I'm going to move on to the final topic in DNA metabolism, which is DNA recombination. And I'm just going to go over this very briefly and talk about DNA recombination in bacteria. So DNA recombination, hopefully that you guys remember, is that a segment of the DNA can rearrange its location. So we're looking at within a chromosome or from one chromosome to another, segments of the DNA can be relocated. That recombination is involved in many biological processes: repair of DNA, segregation of chromosomes during meiosis, and enhancement of our genetic diversity. So in sexual reproducing organisms, recombinations and mutations are two of the driving forces for evolution. Recombination in co-infecting viral genomes, so genomes that, two different genomes that are infecting the same still, may enhance virulence. So this is also a mechanism used for non-sexual reproducing organisms. So let's talk about the classes of recombination. So we have homologous or general recombination. This is exchange between two DNAs that share a region that has a similar sequence. Site-specific recombination, this is exchange only at a particular sequence. And then DNA transposition, and these are the jumping genes, are short pieces of DNA that can move from one chromosome to the other. The three functions of homologous recombination, and that's the one we're going to focus on, is to assist in DNA repair, to link sister chromosomes in the proper segregation between themselves and new daughter cells, and as a source for DNA exchange and therefore genetic diversity and sexual reproduction. So if we look at the process in bacteria, so we have our replication fork, a DNA nick, so a damage in the DNA. So this replication fork comes and encounters this nick. The replication fork will collapse due to the double strand break. So here causes a double strand break, right? Because we have a nick in one end. This is causing a double strand break. Partial of that 5' end will be degraded at this point. So we go from the double strand break to an uneven double-stranded DNA with one end being degraded. That single-stranded 3' end that's left, so this orangish reddish one, is bound to recombinase and pairs with the complementary sequence in the intact DNA in the other strand. So in essence, it invades the other strand. So we have strand invasion and creates this branch structure. The branch moves to create an X-like structure, and we call that structure the Holiday junction or the Holiday intermediate. So we're having crossing over of one DNA strand into the other and vice versa. That Holiday junction is resolved, and we end up with two new DNA molecules, each with a portion of one strand and a portion of the other. Nucleases and ligases restore the structure of the replication fork, and it can come back there and continue to replicate. So we can see that in bacteria, at least in this case, this DNA replication has assisted in what in essence becomes a double-stranded break in the DNA. And now we've resolved it and had two intact pieces of DNA. Okay, with that, we're going to leave DNA metabolism and move on to RNA metabolism. And when we're talking about RNA metabolism, we're specifically talking about transcription. So let's just remind ourselves about the functions of RNA in the cell. First of that being the messenger RNA. So those are the RNAs that code for the amino acid sequences in the proteins, and that makes all of our makes up all our polypeptides, aka proteins that are found in the cell. So the message that translates between the DNA and to protein. The transfer RNAs are the tRNAs, and they're important for translation, so translating from RNA language into protein language, and they carry the specific amino acids for each of the anticodons. And then ribosomal RNA, or rRNA, that's a big component of the ribosome. Ribonucleic acids can also play a couple of other roles in the cell that we don't understand as much about. That includes microRNAs, and those are thought to regulate gene expression, and ribozymes, or RNA molecules that can act as enzymes. So those are two other roles that we don't know a whole lot about, but are new fields in the study of RNA. And then in viruses, RNAs can serve as the genetic material. As you might remember, the process of creating RNA from DNA is called transcription. So that's the process that we are going to focus on for the remainder of this talk. So let's specifically talk about transcription and E. coli. So just like we saw with DNA replication, we add nucleotide triphosphates to the 3' end of the growing RNA strand instead of the DNA strand in the case of DNA replication. That growing strand is complementary to the DNA template strand, so that if there is a G in the DNA, there's a C in the RNA, A, and instead of T in the RNA, we have a U. So it's complementary to that DNA strand. This process is synthesized by the enzyme RNA polymerase, and it also involves magnesium ions to help catalyze the reaction. The RNA polymerase will unwind about 17 base pairs of the DNA and covers about 35 base pairs of the DNA. So if we look a little closer at this process, so here is our template strand of our DNA, and here is our growing RNA strand. If you look at this, it looks pretty similar to the structure we saw with DNA polymerase, in that we have these magnesium molecules working with key residues on the polymerase to add an incoming nucleotide triphosphate. So creating that phosphodiester bond between the alpha phosphate of the incoming NTP and the 3' hydroxyl of the growing RNA strand. The sequence of DNA that the RNA polymerase binds to and recognizes to know when to start transcription is called the promoter. Unlike DNA polymerase, a primer is not required for RNA polymerase. The growing end of the new RNA temporarily base pairs with the DNA for about 8 bases and inserts about 50 to 90 nucleotides per second. The DNA duplex unwinds, forming a bubble. Again, it's about 17 base pairs. And RNA polymerase ends up generating positive supercoils ahead, which can be relieved by those topoisomerases. So we can look at this in pictorial form. So the blue represents the DNA. The DNA is unwound here, and the RNA polymerase comes in and adds RNA again from the 3' end. Here is our DNTPs, our nucleotide triphosphates, that come in and are added to the 3' end. Because the DNA is being unwound, there's supercoils that occur both upstream and downstream, and those have to be relieved by topoisomerase. So when we're talking about transcription, we have to look at each strand of the DNA in a different light. So we have the template strand and the coding strand. The template strand represents the strand of DNA that RNA polymerase binds to and uses as a template to make RNA. The coding strand is a non-template strand, but has the same sequence of the RNA as the RNA, except where there was T's, there is U's. So if we look below here, we have our template strand. That's the strand that DNA or RNA polymerase binds to and makes a complementary RNA to, and then our non-template strand that looks similar to our RNA. So if we look, we have a C here and a C here, a G, a G. If we have a T here, we have a U here. So the coding strand or the non-template strand looks identical to the RNA, except in the RNA we have the U's instead of T's. Let's look a little bit more closely at RNA polymerase. So this is RNA polymerase in bacteria. It's a large enzyme, and it has no proofreading capacity, so it cannot look back and see if it made mistakes. The holoenzyme has 5 core subunits, so 2 alphas, a beta, beta prime, an omega, and the sigma subunit. And hopefully some of you remember from genetics that the sigma subunit is responsible for binding to the promoter. So we have our 2 alphas, beta, beta prime, and omega. RNA polymerase lacks that 3' to 5' exonuclease, so it does have a high error rate between 4 and 5, 1 over 10 to the 4th to 1 over 10 to the 5th error rate. The 2 alpha subunits of RNA polymerase function in assembly and binding to the upstream regions of the promoter. The beta subunit is the main catalytic subunit, so that's the one responsible for adding the new nucleotides. Beta prime is responsible for binding to the DNA. The sigma directs the enzyme to the promoter, and there are different types of sigma subunits that I'll go over in a second. And then omega appears to protect the polymerase from any sort of denaturing. So the promoter sequences in E. coli are really well known. They are they're two consensus sequences, and they're located at the -10 and the -35 region upstream of the start site of transcription. And that is the location of binding of the sigma subunit. And we call those sequences the Tata box or the Tata sequence because they tend to have a lot of T's and A's. These sequences govern the efficiency of the RNA polymerase binding, and therefore they affect gene expression levels. And like I mentioned, nucleotides before the first nucleotide of the RNA molecule are considered upstream and then give negative values. So we count backwards from the start site of transcription, which I'll designate with this arrow. Start site is considered +1. We count 10 nucleotides back, we call that -10. We count 25 back from there, we call that -35. Oh, and I so can't write with that little pen. So here's some examples of those promoters. So we have the -35, the -10, and then the upstream elements that also aid in binding. So for those of you who remember the lovely tripoperon and the lovely lacoperon, again, they have these -35 and -10 sequences. So let's go into the process of transcription in bacteria. So we're going to look here at the DNA, and this is double-stranded DNA. We have our promoter region here, so that's the region where the sigma 70 or the sigma unit binds to the DNA. So here is our sigma factor here that binds to the promoter. The sigma factor recruits RNA polymerase to the DNA after it binds. I guess with this little thing that looks like a foot here, helps bind the RNA polymerase to the DNA. Then it begins to unwind the DNA, and the DNA forms this open complex here, and the RNA polymerase is able to start adding RNA, which is that green molecule right here. So the RNA polymerase will move down the DNA, reading it one base at a time, adding RNA nucleotides to base pair, and it will continue until it reaches the terminator sequence. After the RNA polymerase has started making DNA, the sigma factor is replaced by NUS-A. So the sigma factor disassociates and NUS-A replaces it, and that will go until RNA polymerase reaches the termination sequence. Okay. So as I mentioned, there are different sigma factors in bacterial cells depending on what type of genes they're turning on and off. Sigma 70, which is the one we just saw on the previous slide, is the one we call the housekeeping sigma factor, and that's for all those housekeeping genes. So you can think about housekeeping genes being anything for normal cellular function of the bacteria. So genes for making components of the ribosome, genes for making components for RNA polymerase, things like that. And then we have different sigma factors for different phases in the bacteria's life. The phase we call stationary phase, where it is not actively growing, that's sigma 38. We have the one involved if the bacteria are in any sort of shock, we call that sigma 32. So depending on their functions, there's different sets of sigma factors that are important for different types of genes. So as I mentioned, the RNA polymerase will go through the DNA until it reaches the termination sequence. And there's two types of termination that happen in bacteria. One of them is called Rho independent, and the other one is called Rho dependent. So that little Greek letter is Rho. These are characterized so these are the row-independent ones, are characterized by 3 uracils near the 3' end of transcription. So RNA polymerase is moving down the DNA. It reaches a part of the DNA where there's a large number of T's, and then so it adds a large number of U's into the RNA. Once this RNA is made, this U structure is able to create this hairpin here, and by forming this hairpin, it helps the DNA polymerase release the RNA transcripts. So it forces the RNA polymerase to pause, the RNA polymerase releases the mRNA transcript, and the transcription is terminated. So this is considered row-independent. The second kind is conveniently called row-dependent termination, and it's called row-dependent because it requires a protein called the Rho protein, and if you want to know how to spell Rho, it's RHO. So this is a protein that helps disassociate the RNA from the bacteria. And it binds to a specific site in the RNA called the RUT site, that's that little purple site right there, and is able to move along the mRNA until it hits the RNA polymerase and causes it to disassociate. So row-dependent and row-independent. So in eukaryotes, there are actually 3 different RNA polymerases, and they do different jobs depending on what genes they transcribe. So RNA polymerase 1 synthesizes pre-ribosomal RNA, so that's the RNA important for the subunits of the ribosome. RNA polymerase 2 is the primary one involved in synthesis of messenger RNA. It's a very fast polymerase, but it can be also inhibited by some toxins, including one that's involved in psychedelic mushrooms. And then it recognizes a lot of different promoters, and you can take a look at that figure in your book for the promoters. Finally, RNA polymerase 3 makes tRNAs and some other small RNA products. In plants, there seems to be another polymerase, and that synthesizes for what we call the interfering or iRNAs. And then mitochondria have their own RNA polymerase. So this breaks down transcription by RNA polymerase 2 in eukaryotes. And we talked about the sigma factor as far of helping the RNA polymerase in bacteria. Well, in eukaryotes, the process is quite more complex. And we have a set of different proteins, including the TFP or TATA binding protein, transcription factor 2A, 2B, 2D, 2E, 2F, and 2H. And they all do help in the process of binding to the DNA and unwinding it so that RNA polymerase 2 can come in there and make messenger RNA. So again, we have a TATA binding region at -30, but instead of the 1 sigma factor, there's this group of transcription factors that help assemble the RNA polymerase onto the DNA. Just a more complicated factor than happens in bacteria, as eukaryotes normally are. In eukaryotes, once the messenger RNA is made, it needs to be processed before it can be translated. In bacteria, the translation happens at the same time as transcription, so there's no post-transcriptional modifications that occur. The processes include splicing out portions of the RNA called introns and rejoining the rest of the RNA, which are called exons, adding a cap to the 5' end, adding a poly A tail, and then any potential degradation. So this is an overview of that process. The DNA is transcribed by DNA polymerase 2. A cap is added to the 5' end. The introns are removed, so the yellow portions, and the exons are spliced together, and then a poly A tail is added to the 3' end. The cap, the 5' cap, is a 7-methylguanosine cap, and if we look at the methylguanosine here, it's add to the 5' end of the molecule. The function of that 5' cap is thought to be protecting the RNA from degradation by nucleases. It also forms a binding site for the ribosome. So the ribosome, once translation occurs, binds to this 5' cap and begins translation. The second modification that's made to eukaryotic mRNA after it is coming off the RNA polymerase is the removal of introns. So most vertebrates and some yeast, as well as a few bacteria, have introns within their messenger RNA. So the introns are the portion of the messenger RNA that are removed. The exons are the parts that remain. Exons are usually less than 1,000 base pairs in length, where introns are about 50 to 7,000 base pairs in length, 1,800 base pairs on average. Some genes have dozens of introns. In the human genome, has more than 200,000 introns spread across about 20,000 genes. So there are 4 main classes of introns. Group 1 and Group 2 introns are what known as self-splicing introns. They don't require any additional proteins or ATP for them to be spliced. They're in nuclear, mitochondria, and chloroplast genomes, and the 1 and 2 mainly differ in the splicing mechanism. Spliceosome introns are spliced by an enormous complex called a spliceosome. They're the most common introns, and they're frequent in protein coding regions of the eukaryotic genomes. The fourth class of introns, I didn't put it on here, is in tRNAs, and we're not going to talk about those. So the splicing of group 1 introns. So we have our primary transcript. That's the transcript that came off the messenger RNA. And you can see here's our exon on one side there in green and your exon on the other side. And we're trying to remove this intron portion of the messenger RNA. So the 3' hydroxyl of the free guanosine is used as a nucleophile. And that's represented here. It attacks the phosphodiester bond between the U and the A at the junction between the exon and the intron. So this is supposed to represent a uracil and an adenine, and that P right there is supposed to be the phosphodiester bond. It releases the first portion of the intron, so this portion here, and it in the exon ends with the uracil right here. Okay. That 3' hydroxyl of the uracil on the exon then attacks the 5' guanine on the other exon. So if you look at that mechanism in more detail, here is the uracil and then the adenine. This is the exon on this side, this is the intron on this side, and this is that phosphodiester bond that links them two together. So this is the free guanosine with that free 3' hydroxyl, and that adds as a nucleophile to that phosphate. And it ends up leaving a free uracil and then the guanine attached to the adenine. The second group of introns are the group 2 introns. So we have our primary transcript here with our exon on one side, our exon on the other side, and then our intron in the middle. The nucleophile in this case is the adenine residue and that, 3' hydroxyl, the adenine residue within the intron, which attacks the phosphodiester bond between the uracil and the guanine residue there. After that first cleavage, there is a formation of this lariat structure you can see here. And if you want to look a little bit more closely, we have a 2,5 phosphodiester bond that occurs between the guanine, so that guanine there, and the adenine, which was the one that had the nucleophilic attack. So that forms the the second phosphate on adenine, where the hydroxyl is attached to the 5' of the guanine residue. The rest of the spliceosome is similar to what we saw in group 1, where we have the nucleophilic attack by the uracil to the other side of the exon. The spliceosome is made up of small ribonuclear proteins, or SNRPs. SNRP RNA is also called snRNA for small nuclear RNA, which are about 100 to 200 nucleotides long. 5 snRNAs known in eukaryotes are U1, U2, U4, U5, and U6. The guanine uracil at a 5' end and the adenine uracil at a 3' end usually mark a site of splicing. So if we look at our messenger RNA, we have our exon here and the other side of our exon here. So we have a GAGU and the AGGU on the other side. So the binding of U1 and U2 bind to complementary regions on the messenger RNA. So we see U1 here, we have complementary message. We have U2 here that has complementary to the messenger RNA. U1 helps define the 5' splice site, so the splice site on this side, where U2 is near the 3' end of the intron, so this side. It creates a bulge that partially displaces the active adenine to be a better nucleophile. This adenine forms the 2', 5' phosphodiester bond of the lariat-like intermediate. So the same structure that we saw before, where we create a lariat. So we have binding of U1 to the 5' end and U2 at the 3' end. So this just gives you a different representation of that. So here's U1, and U2. Those are have that RNA within it. Next, U2, U4, U5, and U6 bind. So these are the additional spliceosome proteins, bringing in at least 50 more to create the entire spliceosome. ATP is required for this assembly, but it's not required for the cleavage of the messenger RNA. Some parts are attached to the carboxyl terminus domain, or the CTD domain of RNA polymerase II, so some of them will be associated with the RNA polymerase. This indicates that splicing is coordinated with transcription because these proteins are binding to the RNA polymerase. So in eukaryotes, we also have a process called alternate splicing. So we saw the possible mechanisms for removing the introns and bringing the exons together. But depending on how the exons are arranged, we can get different final messenger RNAs. So one single gene could yield different polypeptides depending on this RNA processing via splicing. So particular regions may be retained, removed, yielding different mature transcripts. So they use some pretty pictures here, but we have the line DNA and the hash DNA and the circular DNA, and it makes those same types of messenger RNA in the primary transcript. And depending on how these are sliced, you can have those 3 exons together or only 2 of them, etc. So we can get 2 different mature mRNAs from one DNA molecule. and we call that alternative splicing. At least 95% of the human genes have alternative splicing. And the final thing that happens to the messenger RNA during processing is the addition of the poly A tail, and that's multiple adenines added at the end, the 3' end of the messenger RNA. That serves as a binding site and it helps protect the messenger RNA from degradation. So the RNA polymerase II synthesizes the RNA beyond a cleavage site, and the cleavage site is bound by an endonuclease and the polyadenylate polymerase that's going to be responsible for adding those adenylene residues at the end. The endonuclease cleaves the RNA within 10 to 30 nucleotides downstream of this sequence called the AAUAA sequence here. And then polyadenylate polymerase synthesizes 80 to 250 nucleotides of adenine. And you can see that N here means it can be anywhere between 80 and 250 nucleotides. So to wrap up RNA processing, just give you a brief summary of the whole process. So the process of transcription is taking DNA and making it into RNA. And in eukaryotes, this is called a primary transcript. This process is catalyzed by RNA polymerase II, which is different from what happens in bacteria. This primary transcript gets a 5' methyl guanine cap here at the 5' end, and this is the binding site for the ribosome. It undergoes the process of splicing where the introns are removed and the exons are brought together. And then at the very end, we have the addition of the 3' end of the poly A tail, and that's catalyzed by the polyadenylate polymerase. So that's the general overview that happens in eukaryotes. And hopefully, as you remember, this is not what happens in bacteria. Bacteria goes straight from DNA to RNA to ribosome. There is no processing that happens in between. And with that, we're going to end on RNA and come back next week.